Alright, hello everyone. Let's talk about condensation and hydrolysis reactions. So what I want to try to do is bring you back to what you had in your biology class, your freshman or sophomore biology class. This will look familiar here very soon, hopefully. So condensation, we know what this word means right here. So the term condensation, this means kind of to bring together. All right, so when we condense things, all right, so when we condense, that usually means that what we do is we combine. Yes, and I know my handwriting is not the best, so I will bear with me. It will get better as I practice this some more. It means we combine it. Okay. So we're going to have a reaction that will be a process of combining, and then we're going to have a reaction that's going to do the opposite. And so you should hopefully remember that this term from biology class, when you talk about lyse, L-Y-S-E, what that means is to break. So you remember you had like a lysosome, and that was the organelle that was responsible for breaking apart things, old parts of the cell, or things that were contaminated in there, waste materials and stuff like that, so that the cell could get rid of that stuff. And so hydro obviously kind of makes us think of water, right? So both of these reactions are essentially what happens with water on whether on molecules being combined or being broken apart. Okay, so that's first and foremost, that's what we need to get out of this. All right, so let me go ahead and erase all that. And we've been studying things like macromolecules, right? And so before this particular video here, one of the things we've been talking about in class were macromolecules, right? So we've got our macromolecules. And the macromolecules that we've talked about are things like amino acids. Well, amino acids we said were the monomers. So let me back up. So the macromolecules, like we had proteins, right? And then we had said our macromolecules were made up of monomer units. And for proteins, we know that that's amino acids. All right, we had lipids. And remember we said glycerol, and then we had fatty acids. And then we had our carbohydrates. So here's where you've heard the term like complex carbohydrates or complex carbs or carbs, right? And then, of course, from biology class, you remember, and we discussed that glucose was our monomer unit. Okay, so from these things here, what we want to do is take a look. First, we'll take a look at proteins, then we'll take a look at lipids, and then carbohydrates. Okay. So let me go ahead and let me get rid of all this stuff here. Let me get rid of this. Make some room on the screen for you. All right. You know, at any time, you can always pause and stop the video, go back, take notes. You know, this is your video to do with what you need to get prepared. All right, so let's look at the condensation. All right, if we look at condensation reactions, what we're going to find is once we understand this reaction here, that a hydrolysis reaction is literally just the reaction in reverse, or the reverse of it, I should say. So you in biology class you studied like cellular respiration and photosynthesis, and you talked about how the products of one reaction were the reactants of the other, and it sort of circled back on itself. So we're going to find here that in a condensation reaction, when we're done, whatever we'd have on the product side If we thought of those as being the reactants, then the reverse of that would give us the products, which would be these reactants. And we'll see that after I get done. So let's take a look at condensation. All right. So let's look at proteins. So we've already said that proteins, we know we need amino acids, right? And so we know our basic amino acid it had this functional group, this amino functional group, right? And then there was a carbon here, right? To another carbon, and then remember we had our carboxylic acid group. And then there's some R group. We don't know what it is, we just know that it's some group of atoms. It means like the rest of the molecule. But this part right here, this part doesn't change. That was pretty consistent, right? We know that this is our hydrogen right here on this part of this hydroxyl group. So condensation means, remember we said this is combining. So what happens is here's a monomer unit. Well, if there was another monomer unit here, so let's take another amino acid. Okay. Another amino acid. And again, all the basic parts are there. It's just the only thing that makes this amino acid different is whatever the R group is. Remember, the simplest R group is a hydrogen atom, and then the largest R group is just, it can be a ringed structure, it can be a bunch of atoms, all right? But this main part here is never going to change, the amino part and our organic acid part. That's why it's called an amino acid. Okay, so what happens is in a condensation reaction is there are parts that sort of interact. And so the hydrogen from this part of this amino group and the hydroxyl part of this carboxylic acid group, they interact. And you should be able to see that this is H2O. And so what happens is water comes out of it. So I'm writing HOH. This is kind of like the organic way of writing water. You can write H2O, it doesn't make a difference. It's still the same thing. So I'll write it this way sometimes. Sometimes I'll write it H2O. It's still the same thing. So you should see right here there's a reaction. All right. Well, let's think about this for a second. All right. I'm going to use an eraser and just temporarily, if we know that this reacted right here, then that means that those two atoms, they're gone. They're not there anymore. And so now you should be able to see that right here, this carbon atom would have one free electron on it, and this nitrogen over here would have one free electron, which means that this carbon atom here and this nitrogen atom here, they could combine. They could bond together, and there would be a bond between that carbon and that nitrogen right there. So let's do that. So I'm going to draw an arrow, okay, and real quick, what I'm going to do is just give us a little bit more room here real quick, okay. So right here, remember, this is the part that reacted. So you don't have to change any of this. Everything right here, every bit of this. So right here, only what's highlighted is what's going to change. Everything else stays the same. So if it's not highlighted, if we haven't encircled it in some way, if we haven't identified it in some way, leave it alone. So all you got to do is rewrite it. So that means we've got this amino part over here. And again, what I'm doing right here is I'm redrawing this part. I'm going to draw all of this right there. None of that has changed. Okay, so it goes to a carbon, and then we see it's at a hydrogen. There's that R group, and then there's this carbon. This carbon I just wrote is this one right here. So now real quick, before I go any further, let's think about this. This carbon is bonded to this nitrogen. All right, so I'll change the color just so you can see it. But again, oh, that's pretty bad. So I'm changing the color just for training purposes, just because I want you to be able to see the difference. If you think about this, you see how we've talked about angles. So remember how this is 1, 2, 3 bonding regions right here, right? And so we know the angle that this would take. Remember this from our molecule kits. You see how this double bonded oxygen is coming out at an angle, kind of in this direction, and you can see how this nitrogen is coming out at an angle this way, right? We've almost got those trigonal planar sort of angles going on. If these two parts get closer together, and we know that right here in this region of space, and the two lone pairs that are on this oxygen, because remember we can't forget those, if they come close together, then like we've already been studying, the electrons here are going to repel each other, which means that this bond right here, this right here is going to move over more this way where it almost be straight. And so would this nitrogen here. Instead of being up this angle, it would be straight. So this carbon here is this one. So I'm going to draw this going straight up. This carbon is now bonded to this nitrogen. This nitrogen bonded to that hydrogen, and so we said that hydrogen would go straight up. And now you literally just redraw the rest of the molecule. And so this part right here that I'm doing right now, that should not be too much of a challenge for you, because I have asked you on your own to be practicing all this. And over here I put the R in a box. I just did that to highlight it for you. You don't need to do that. I'm just doing that in this video so that you can see the parts. So now, right here, we can see right there that we got a bond that formed. And this right here, your biology teachers called this a peptide bond. Okay? That is a peptide bond. You'll remember that they called proteins polypeptides. And so right here, this specifically, we would call this a dipeptide, because there are two amino acids that are linked together into this one larger molecule. We essentially condensed these two parts right here. We condensed this amino acid and this amino acid together by removing water. When that happened, the carbon and nitrogen bonded. So this carbon-nitrogen bond is called a peptide bond. And now we call this larger macromolecule a dipeptide. All right. In biology class, you called condensation. The term for this that you also had was dehydration synthesis. Okay. And the reason for that was, think about this. If you're dehydrated, that means you feel like you've lost some water. You feel like you need water. Well, if you're dehydrated, that means you've lost water. So let's look at this process right here. When these two things came together, water had to be removed from what was combining. And synthesis means a process of putting together. If you synthesize something, you are making it, you're putting it together. So this term dehydration synthesis makes sense. When we take water out, a process of putting things together, by taking water out, we condense them, which is why it's also called condensation. Okay? So now, if I were to ask you, let's just say we had never talked about this before, and I were to ask you to write the condensation reaction for a dipeptide, this is exactly what you would show. You would have to show this amino acid, this amino acid. You would have to show that this part of this functional group and this part of this functional group chemically come together, water is removed, you show your reaction arrow, and in biology class, what you typically did as well, above the arrow, you would write something like enzymes. because enzymes were basically your organic catalyst that brought it all together. Oftentimes enzymes or heat, or there has to be something, there's usually something that will catalyze these organic molecules. So here's your dipeptide. So there's the condensation of the to make a very simple protein. All right, so that's one down. Let's move to lipids. All right, let's move to lipids. So I'm going to let me highlight all this. I'll get rid of all that. Let's make some space here. And now let's go with the condensation of lipids. Actually, real quick before I do anything, let me go back to what we had just done. Before I forget, we also have this part here about hydrolysis. So this is what I need you to be able to visualize. Okay, this is what you have to be able to visualize. You have to be able to visualize that. So we know that this is our reactant side. These are our reactants, and this dipeptide here, this is our product. And in a chemical reaction, you have reactants that make your products. If we wrote this on the reactant side, then we write our arrow, and then we would show both of these here on the product side. That would be hydrolysis. Now there is one thing I missed here, and I am notorious for this. I always miss this. One thing we miss in this reaction is to add water on this side. So let's think about why. We have this amino acid, they came together and water came out. So if we think of what you learned in your basic chemistry, law of conservation of mass, all these atoms here, we have to account for them. So if this hydrogen, this oxygen had been removed in the form of water, then that means it's over here, plus we now have all of this that was remaining that was chemically bonded together. So now we have a reaction that kind of shows the law of conservation of mass. Okay? And so again, if I were to write this on the reactant side, and then if I were to put this dipeptide and water over here on the reactant side, and then I were to put all of this on the product side, that would be the hydrolysis reaction. Okay? All right. So let's do this. Let me go ahead and get rid of all this here again. Just make some room. All right. Let me erase this for you. Remember, at any time, you can pause and stop and go back to anything that we're doing. All right, so let's look at lipids. All right, so with a lipid, we know we have, with a lipid molecule, we know that we're going to need glycerol plus a fatty acid, right? So remember from our studies that glycerol was this 3-carbon chain, and these 3 hydroxyl groups were attached to it. So remember, we remember from glycerol, we know glycerol is basically a type of organic alcohol. It's an alcohol, right? Because of those hydroxyl groups. So we've got our glycerol right here. Okay. Here's our glycerol. And then we need to add a fatty acid. So remember our fatty acid, so the word acid and the fact that we're in organic chemistry, every time we see this term, you're going to need a carboxylic acid. So there's our carboxylic acid. And in class, we've already taken a look at this. We know that there's a bunch of CH2s. How many? We don't know, so we just put the N. And then we have this methyl group that's at the end. So remember, if N equals like 4, then we would put 4 CH2s and we would stretch that out, right? Okay. So let's think about this for a second. this thing here. So I know there's a shadow effect there, but I'm going to pick this up and I'm going to move this kind of somewhere. I'm going to move it, stay right there. We know, oops, sorry about that. We know that what we need is water to come out of this. If we're going to have a condensation reaction, if we're going to have a condensation reaction, that means these two parts have to come together. And that means we know from our previous studies that dehydration synthesis is what's occurring. So it means we're going to have to lose some water. Well, we see oxygen and hydrogen, and then oxygen and hydrogen. Well, we only need two hydrogens and an oxygen. The acid part is very reactive. So what I'm going to do is just kind of draw some little dotted lines around there. And from right there, that is where our reaction is occurring. From here, this is where water is being removed in this reaction. Okay? So now let's think about this. Oops. That was pretty bad. So let's think about this. Everything that is not highlighted did not change. So all that means is I have to worry about just joining what is highlighted together. So let's use our eraser again. All of these atoms, if they're removed in the form of water, then that means now this oxygen here has to chemically combine with that carbon there, because now there's open space, so to speak. Remember, we got those free electrons. Remember, we have basically just one electron left there. Because remember, these are covalent bonds. So anytime things are removed, those bonds, part of the bond is removed. That means there's electrons that are available to join up, to bond together. So there's going to be a bond between this oxygen and that carbon atom right there. Okay. So let's do that. Let's get this combined. So that means, remember, all of this stays the same. All of that stays the same, and we're just going to want to combine from this oxygen to that carbon. So what I need to do is draw what I know did not change. All you're doing is recopying what you already know. And if you've been practicing all this, then it's really not too challenging. The only new part here should be trying to wrap your mind around the fact that once this is gone, the bond is there. If you're practicing drawing each of these monomer units, then this is really not too terribly difficult. So now we've got this carbon bonded to an oxygen. That oxygen did not go away. And then this oxygen, remember we said, has to bond to that carbon. And remember, if we bring this entire molecule over to here, and you see how this is coming out at an angle, it wouldn't make sense. It would not make sense for that to stay at that angle, because right here there would be that repulsion as it gets closer. Because remember, there's lone pairs right here, so it makes sense that this would move more in this direction. This would be pushed, it'd be repelled in that direction. So right here, I'm going to write that double bonded oxygen, kind of going vertically like that. And now all I'm doing is just copying the rest of this. That's a carbon, and we have our CH2. We don't know how many, so that's why I'm writing an N. Okay. So now right here, there's that bond between that oxygen and that carbon right there. You should be able to recognize now, okay? I'm going to actually go back to the highlighter, but you should now be able to recognize that all of this right here, you should now be able to recognize that once this is all bonded together, that this whole thing that is highlighted is an ester bond. And like an ester functional group. I'm sorry. Let me. It's the ester functional group, which we would sometime then because this ester right here is linking this and this, then we sometimes call it an ester linkage. Okay. This part right here, remember this bond is what formed right there. This right here is this is our bonding site that ends up forming this entire ester linkage. And so you should be able to see now that there's a hydroxyl group here that if a second fatty acid were to come right here and join, you would get another bond right there. You would get another one of these ester links, and then you would have something like this here and something like this here. And with this hydroxyl group here, I forgot to write that hydrogen there, sorry. You would also get another one of those. So it's possible for 3 fatty acids to bond to one of these glycerol molecules. All right, and that'll be a lesson that we'll look at that more clearly in another video lesson. All right, and in class as well. All right, so hopefully that makes sense. All right, let's move on to the very last one. We have one more to do, one more of these. And we want to look at our sugar. All right. There. So let's look at our sugar molecules. And so we know for that we were using glucose, right? Okay. So let's think about this for a second. Hopefully you've been practicing these. There's a lot here. There's a lot to worry about. But you have to be able to draw the basics of everything. All right? That was not very attractive. All right, let's see, H, and then we have an OH. Imagine that this entire part here folds over like a book. Comes over here, and there's an OH here, and this is an H, OH, and then we have OH. Those hydrogens are pretty ugly, oh my gosh. Sorry about that, but luckily I can undo it and then take my time. So there's our glucose. And remember we said this was the alpha glucose. So this reaction that I'm going to show you, it happens with the alpha glucose, it happens with the beta glucose, but the alpha glucose is essentially the one we use just as an example, because that's the one we talked about in biology, and it does happen a lot. Your body uses this primarily for many reactions. Okay, so this is a condensation reaction. Condensation means you've got to add them together. So I need another one of these things here, right? All right, so let's go ahead and draw this out. So we've got another one of these, and I know I could probably just go copy and paste, but since you have to write it out, I'm going to go ahead and write it out as well. Okay. So we've got CH2, OH. Here's a hydrogen. Here's a hydrogen with our hydroxyl group. Hydrogen, hydroxyl group, hydrogen, hydroxyl group. It comes over on this side, it rotates up, and it bonds that particular way. And here's another alpha glucose. You should be able to see that those two right there, that's where we're going to have a reaction site. Okay. And right here is where water comes out. Before I forget, I should note that on the previous part of this lesson. I forgot to write the water when it came out for the lipid again. So make a note. Yes, I did forget that, and I have said this is one of the things that I tend to kind of do. I sometimes forget to write the water. The water has to be there. Remember, law of conservation of mass. I'll make sure to write it on this one. Okay. All right. So we've got these two sugar units, right? We got these two monomer units. I'm going to go ahead and make that a little bit smaller, and I'm going to move it right here to the center. It's not changing anything different. This molecule is large, and I have enough space probably to go in this direction here. But I'm going to just use this opportunity to show you that you can. If you don't have enough space to write left to right, you can go top to bottom with your reaction, and whatever's on top would be your reactants, and whatever's the arrow's pointing to on the bottom would be your products. All right. So again, we see, if we use our eraser, let's envision that those atoms have been removed. You can see that there's an oxygen right there that's going to have to swing up and bond right there. Remember, each point is a carbon. So the oxygen here is going to bond to that carbon right there. Okay. So that means all of this, every bit of this right here, every bit of this is going to everything that's here that's not highlighted, it just stays the same. So we just have to go through the laborious task of drawing it all over again. All right. Every part that did not react. What I'm going to do first, though, is I'm going to bond this together with since we don't have to worry about all this stuff here, let's just draw the parts that we need to worry about first. We see that this carbon is that carbon there, and there's an oxygen here that did not go away. And this oxygen here is going to swing up and bond to this carbon point right here. So that means I just need the rest of that glucose molecule. All of this other stuff, all these other hydrogens and hydroxyl groups, they're all there. All I want to do is just highlight what we know has occurred as far as the reaction goes. That is what is left. Okay, I'll get the rest of this stuff here in a minute. When two sugars come together and bond, this bond that is here, we call it a glycosidic bond. And the glyco part, that is a prefix that means sugar. It is a prefix that means sugar. All right. So now, remember, this water came out. So what else do we have here? We also have some water. All right. Now we just go back. Oops. All right. We just go back. I'll try to be consistent with my colors for you. We just go back and we're going to draw in everything that did not change in the reaction. So now we have all of this here. And again, if this is more confusing if you are not used to drawing these molecules. If drawing these molecules is still a struggle, then this is going to seem incredibly difficult. You have to get used to drawing the molecules. Once you do that, this is pretty basic. It just takes practice, which I've given you some practice in class. All right, you have practice problems for this, etc., etc., etc. All right, so now this hydrogen here didn't react, so that hydrogen there, it's still there. Right here, that oxygen is that one is taken care of. So now we just have to put the rest of this in, so we know we have a hydrogen. We got that there. And then we go back here, we just put everything else that is there. That's carbon, CH2OH. There's an H there. this hydrogen here didn't go away. And I'm drawing them kind of straighter because remember there's lone pairs here, so they're at an angle here, but they would be kind of pushed away as these two atoms, as these two molecules get closer together, just like there was in the previous one. All right, so we're literally just, and I'm going to have to erase this part here. I'll rewrite that later so that we can write all of this right here. So we've got our hydrogen and our hydroxyl groups. And remember, yeah, this is still our glycosidic bond. Okay. It's balanced out because we see here we have this monomer unit, this sugar. We have this sugar, very simple sugar, glucose. Both of these are alpha glucose. And through this condensation, it's been dehydrated to synthesize this one larger molecule here. All right. And now we have our balanced reaction. Hopefully this makes some sense. If you don't forget, there's online things that show this, like the gizmo that I've assigned. You can it actually uses this exact reaction right here. And again, the opposite of this, the hydrolysis of this, would be this larger, this molecule here, which is two sugars together, and your biology teachers called these two, when you got two sugars that are bonded together, they called it a disaccharide. So a polysaccharide was a large sugar, right, et cetera, et cetera, et cetera. So here we have our disaccharide. If I wrote the disaccharide plus water on the reactant side, and then I wrote these two monomer units, these two alpha glucose on the product side, that would be hydrolysis. That's the only difference, is where the actual molecules are. Okay, what's happening with the reaction. So if it was a hydrolysis reaction, this water molecule would come in here, it would break this bond and separate these two out, where you would then still have OH here and then OH here, like you've got up there, and you have separate molecules. You would have your two monomer units. All right, so feel free to pause, rewind, and go back over this. Take plenty of notes. And again, to make this less confusing, if you haven't done so, you should be going back and just practicing writing your individual monomer units, and then at that point, it doesn't matter if we have two of these together or 200 of them together. It's just the same process over and over and over again. And I'll make a video about that later. Hope this helped.